All right, let's jump right into this explainer because what we're looking at today is, well, it's a truly mind-bending journey into our own DNA. We're going to explore this emerging field called paleovirology and uncover a biological plot twist that completely rewrites how we think about human evolution. You know, we usually consider viruses to be these deadly pathogens, right? Invaders that just make us sick. But what if these microscopic invaders might actually be the very reason we exist at all?
It brings us to this ultimate biological paradox. I mean, think about this. What if the very thing that makes us mammalian is actually a virus? Today, we're questioning what it really means to be a placental mammal. Because if you zoom back through millions of years of evolution, it turns out that our survival, our ability to even reproduce, depends entirely on ancient captured viral code. Let's trace exactly how this happened.
Section 1. The viral DNA within us. To wrap our heads around this, we have to recognize that our DNA is essentially a microscopic archaeological dig site. It's totally full of fossils, but instead of dinosaur bones, it's packed with ancient viral genomes. And look at the sheer massive scale of these ancient invasions. 45%, literally 45% of human DNA and about 40% of mouse DNA are made up of endogenous viral elements and other ancient genetic fragments. Now, for the longest time, scientists just completely dismissed this massive chunk of our genome as mere junk DNA. But, as we're going to see, it is far, far from junk.
So the crucial point here is understanding what exactly is lurking in our DNA. We're talking specifically about endogenous retroviruses, or ERVs. An ERV is basically a fossilized retrovirus that didn't just infect a random cell, it infected a host's germline, meaning an egg or a sperm cell. It integrated its own code directly into the host's genome. And because it was in a reproductive cell, it got passed down vertically to every single offspring over millions of years. So these aren't just any viruses. They're viruses that got permanently trapped in our ancestors' reproductive code.
Section 2. How viruses invade germlines.
So how does a virus actually become a permanent part of our genetic blueprint? It happens through this fascinating mechanism called endogenization. We can see a step-by-step breakdown of the process right here. Normally, retroviruses have a pretty simple genome with three main genes, GAG, POL, and ENV. When a retrovirus infects a somatic cell, say a lung cell, well, that infection just dies with the host. But look at step 1 here. An exogenous retrovirus infects a germline cell. Moving to step 2, it converts its RNA into DNA and integrates that proviral DNA right into the host's genome. And finally, step 3 is vertical transmission. Because it's embedded in the reproductive cells, the host now passes those viral GAG, pollen, and genes on forever.
And this right here is the exact moment of domestication. Once the virus is integrated into the germline, every single descendant inherits this viral code in all of their somatic and germline cells. The virus has effectively been domesticated by the host species. Crazy, right?
Section 3. The envelope protein secret.
Now, over millions of years, most of these trapped viral genes just mutate into completely useless fragments. But our ancestors kept one very specific part, the retroviral envelope protein, which is encoded by the ENV gene. This protein essentially has superpowers. First, it features a transmembrane subunit containing a fusion peptide. This is what allows the virus to fuse its membrane with a host cell just to get inside. But even more fascinating? It has an immunosuppressive domain, or ISD. This acts like a literal invisibility cloak, repressing the host's antiviral immune response so the virus doesn't get destroyed.
So normally, the virus uses this envelope protein to fuse a virion into a single cell. But look at what happens when infected host cells themselves express this viral protein on their surface. They actually gain the ability to fuse with adjacent uninfected cells. This creates these massive multinucleated giant cells called syncytia. It's a mechanical fusion effector that was originally designed for viral entry, but now it's causing host cells to literally melt together.
Section 4. Building the human placenta.
All right, here is where the massive scientific plot twist happens. Our mammalian ancestors hijacked this exact cell-fusing, immune-suppressing viral technology to solve the ultimate biological problem, pregnancy. To understand why this was even necessary, we have to look at something called the fetal allograft problem. When an early mammalian ancestor evolved to carry its young internally rather than laying eggs, it faced a massive immune challenge. You see, a fetus expresses foreign paternal antigens, so medically speaking, it's essentially a foreign tissue graft. So the huge question is, how does a mother carry this growing fetus without her immune system immediately attacking and rejecting it? Well, the answer required a highly specialized, highly tolerant barrier.
And the evolutionary solution was the placenta. Specifically, the placenta features an outer layer called the syncytiotrophoblasts. This layer mediates nutrient exchange and provides this crucial environment of immune tolerance that totally protects the fetus. But look at its structure. It is a highly invasive, multinucleated syncytial layer. It's built entirely by the continuous fusion of underlying single cells called cytotrophoblasts. Now, does that fusion mechanism sound familiar to you?
Well, we have direct genetic proof of this connection. Scientists found two specific human genes, syncytin 1, which is about 30 million years old, and syncytin 2, which is over 45 million years old. Listen to this. These are not human genes. They are domesticated retroviruses. And as you can see in the tissue analysis here, syncytin 2 and its receptor MFSD2 are exactly what drive the vital fusion of these placental cells today. We are literally using an ancient virus to build our placentas.
Section 5. Proof from mutant mice.
Now you might be wondering, okay, how do we know this isn't just some wild coincidence? Well, scientists didn't just guess this connection, they proved it experimentally by looking at what happens when these exact viral genes are turned off in mice. Mice also have placentas, but their architecture is just a bit different. While humans have one syncytiotrophoblasts layer, mice have two distinct layers called STI and STI2. And guess what? Mice have their very own ancient domesticated retroviral genes doing the work. They're called syncytin A and syncytin B, and they've been conserved in mouse ancestors for over 25 million years just to build these specific layers.
These knockout studies paint a devastating, yet highly informative picture. When looking at normal cells expressing syncytin, we see typical, healthy fused syncytia. But when researchers actually knocked out the syncytin A gene in mice, the result is shocking. Cell fusion completely stops. Unfused cells accumulate, the early placental architecture is entirely disrupted, and tragically, the mouse embryos die at mid-gestation. It is absolute, undeniable proof that without these viral genes, placental reproduction fails completely.
Section 6. Convergent mammalian evolution.
As if that wasn't wild enough, we are about to hit the most mind-blowing fact of this entire explainer. This incredible viral hijacking, it wasn't just a one-time fluke in evolutionary history. Entirely different animal families independently captured distinct retroviral envelope genes over millions of years. Primates captured syncytin 1 and 2. Mice captured syncytin A and B. Rabbits captured syncytin ori 1 over 12 million years ago. And carnivores, like dogs and cats, they captured syncytin CAR1 over 80 million years ago. This is an absolutely stunning example of convergent evolution across entirely separate branches of the animal kingdom.
So, what this tells us is that as mammals evolved, different ancestral species encountered and domesticated entirely different viruses by sheer chance. They co-opted these new ENV-derived genes, sometimes repeatedly replacing older ones, to continually refine and develop the placenta. Nature literally just used whatever viral building blocks happened to infect their germlines to gain an immense selective survival advantage.
And that brings us to a really provocative final thought. If our very existence as placental mammals relies completely on captured viral code, if we literally cannot be born with the fusion proteins of ancient retroviruses, how human is our DNA really? It completely forces us to fundamentally reconsider the boundaries between human evolution and the ancient pathogens that literally gave birth to us. Thank you for joining me on this incredible journey through our genetic fossil record, and I really hope this completely changes how you look at the natural world.